teach their biochemistry, molecular biology, and medical students, and to give them an idea of how these structures work and what these molecules do. And these are membrane proteins. These are structural proteins up here, giving that cell wall up there some strength.
These are actual...
This is what they look like. These are not just cartoons. We really know what they look like from X-ray studies. This is actin. This is the fiber we talked about. This is what gives strength to the little microvilli that stick out of the cells and things like that. This is how actin is made. It's a monomer that polymerizes, and you can see how it looks like it has ratchets. Okay? This is a protein that can cut it and destroy it. Okay? This is tubulin. These are the microtubules. This is how they're put together. Okay? And they can dissolve like that really fast. And this,
Oh my gosh.
yeah, you say, what is this? That is the most fantastic thing I've seen since I left school. Nobody really knew how these things worked. Nobody really knew what they were. But it turns out that there's a general class of these things called motor proteins. Okay? They have different names, kinins, dinins, whatever. Myosin is one of them. Okay? You'll notice they're almost like antibodies. It's a kind of a combination between an antibody, which usually has two forks like this, and it bonds to receptors on whatever this is. This just happens to be some sort of vesicle or something that this thing is dragging along this microtubule. This is how things move around the cell. Okay? We never knew that when I was in school. Okay? We thought that it was just like diffusion. You know, everything's just bouncing around in there, and you look at an amoeba and you see all these things moving. But we really didn't know that the cells direct where these things go. Okay? And these motor proteins, they can grab onto a mitochondria and drag a mitochondria along a microtubule all the way from the cell body that lives in your spine all the way down to the end of the nerve in your big toe.
Wow.
Okay? And this is how these organelles move around inside the cell. And because of the receptors, there are certain types of motor proteins that will only hook on to certain types of things. There are some motor proteins that will only go in one direction along the microtubule because it's not symmetrical. And there are others that go the other direction. And I've even seen electron micrographs of vesicles zooming up a microtubule, and all of a sudden it's zooming back the other way. So another protein grabbed it and started taking it the other way. But this little walking mechanism here, it's not magic, it's just protein. So, but if you've got all those alpha helixes and beta sheets in the right way, all you need is for an ATP to put a negatively charged phosphate group on one area, and you're going to get repulsion or attraction depending on the electrostatic charges, right? And when you get that, you get tension in the structure. You're bending those bonds and making tension. So then when it leaves, you release that tension and it moves. Okay? So that's how these things are moving and walking. But it's all ATP driven. And it might spoil some of the fun, but when we get to our muscles, you're going to find that we have structures just like this, only instead of 2 feet, we only have 1 foot. But they take hundreds of them, and they wrap them around each other, and now we've got lots of feet walking down that thing. That's how our muscles contract. Okay? So, but this is the coolest thing I've ever seen. So,
question, Dr. Ertz. Every time that thing moves, it uses ATP?
Every time. Each one of those legs is using 1 ATP. So you can see we're making trillions of ATP molecules all the time because that's what runs everything. Okay? That's what slams ATP is what gives charge and what slams the door shut on pores and things like that. We have pores that will let things in, but you put it on charge on it, or put even a sodium positive charge sometimes will shut these things off and on. So we have valves in our pores. It's just incredible micro machinery, but the way these things function is what we're going to be talking about this time, okay, on this test. Okay? But anyway, so I'll show you the rest of this here. There's the centriole making the microtubules. These are pores in the nuclear membrane. They're really pretty good size, and they're letting out RNA. This is the copy of the DNA that's going to be made into protein. There's the ribosome running along the RNA, and the protein is now being formed. They're not showing you all the stuff coming in that forms it, you know. But you're going to learn how all that works. There's one. It's now sitting on the endoplasmic reticulum, and it just made a protein that went inside. Okay? And then you get these vesicles that form off of the endoplasmic reticulum, and they get dragged up to the Golgi apparatus. And then it's going to fuse. See how the vesicles just sort of fuse into the membrane? So all the stuff that was inside is now inside the Golgi.
Right.
The Golgi then forms vesicles and sends them elsewhere. This one part back here is worth seeing again. Okay. Anything inside the vesicle, when it opens to the outside, is going to be released. Anything that's in the membrane of the vesicle is now going to be part of the membrane, the cell membrane. So see, these are stuck in the membrane. They are now membrane proteins. That's how they get there. Okay. This hard area right there is called a raft, and that's usually, it gets stiff because there's a lot of cholesterol in that area. These are cell identification proteins that so the cells know who each other is. That's a white blood cell that just dissolved all its microtubules and actin and everything so it could slide between these two endothelial cells. Okay. So they've done some fantastic stuff, and there's a lot more on the internet if you just look at Harvard BioVisions. But this kind of stuff I think is very teachable. So I haven't watched them all, so I don't want to really waste your time if it's not useful, but there are a lot of good ones.
That video looks relevant. The turtle.
What's relevant?
The turtle. With everything else in there.
Yeah, you gotta wonder. Or they're going to find a raven with a watermelon. We're going to watch things like, you want to see a fantastic molecular machine. I haven't really watched this one, but it's got 66,000 views. We're going to be talking a lot about ATP synthesis. You get hydrogen that's secreted in the membrane, and then it has to flow.
I think we got spoiled by the last one.
Yeah, that last one's too good.
There's no music in this one.
Let me do this. Okay, I'm already.
These 3D renderings are a bit archaic, don't you think?
Synthase is the name of what we're looking for. And let's see if we can't get a decent one here. Yeah. Alrighty. Okay. 26. I've seen this one before. It's a minute long, so I don't want to waste most of your time. This is the motor. This is a protein motor that uses hydrogen ion flowing through the membrane to drive this rotor, which makes these proteins rock back and forth, and that gives the mechanical energy to force the phosphate onto the ADP to make ATP.
Is that what you were showing us last week on that slide where you had the globular protein, or is that different?
Different, yeah.
That's different, okay.
Yeah. This is a really interesting motor. See how that thing is moving? And it's only because it's like hydrogen is flowing through here. It's like water going through a water wheel.
Is that a chaperone protein?
No, but it's certainly folded over the axle. But this rotor is connected to an axle that's off center, so it does this, and it forces these proteins to move, and the movement is what forces the phosphate onto the ADP to make ATP. So it's an incredible process, and that wasn't known how it worked either, okay, when I was in school. So this one is very similar. I think it's just a sort of a top view. Maybe that'll show you. But we'll be talking about this time. So you can see the phosphate and the ADP. As this thing moves, they come in, and it moves again, and then they leave as ATP. That's where they fit, and they have to fit perfectly, and then they get squished together, and then the next rotation it will open up and let the ATP go. See, now we've got a new ATP molecule going away. So you're really using a chemical gradient that you've got. That's the protein. They would be considered, you know, globular type proteins, but they're in the membrane. That's only in the mitochondria. That's why we need mitochondria. That's why we get oxygen to the mitochondria, so they can do this. Okay. It's out there if anybody wants to look for it. I told you I was going to give you a little special 10-minute thing on sexual differentiation. I forgot I didn't give it to this group. Okay, I gave it to the other group. Just a couple of words you might want to at least have heard. One is called virilization. That's when females are masculinized. Okay, this will not be tested. This is PBS, every bit of it. Okay. Everybody's reaching for their notes. And then you've got undervirilization or feminization, which occurs in males. And there are different ways that this can happen. Congenital adrenal hyperplasia is one of the more common ones for females. And you'll notice that it says that due to 21 alpha hydroxylase, okay, what's happening right here, here's the 21 alpha hydroxylase. If your adrenal gland can't make all of your corticosteroids, like aldosterone and cortisol, your pituitary gland says, hey, you know, we're not getting enough of this stuff, you need to make more, and it starts to send hormones to stimulate the adrenal gland of the female, but nothing's happening. And she'll usually be born with an adrenal gland, it might be the size of a cantaloupe, okay? So that's why they call it adrenal hyperplasia, okay? But what she also doesn't have is any of the stress hormones or the hormones like aldosterone that govern potassium and sodium balance, things like that, fluid balances. So she's going to have to be taking a lot of medication during her life to help control all this stuff. But what tends to happen is if you can't send the steroids in that direction, you get way too many of them going down in this direction, and they get synthesized into the male hormones more, okay? So she's born with a lot of extra male hormones in her system, and she's born looking like a male, okay? They will, you know, you couldn't tell just by looking at her when she's born, okay? You'd have to do a genetic testing to see. Another problem that can happen is when females can't make aromatase, okay? Aromatase puts those 3 double bonds in that ring right there and takes that ketone to an alcohol. And that's the only difference between the testosterones, you know, the precursors, and the estrogens, is that aromatic ring there. Okay? So if aromatase is missing, you will be born looking like a male. Okay? Normally, the default development is female. Okay? If you don't have any male hormones, you'll be born looking like a female. Okay? Or if you're born with a disease where you have receptors that aren't responsive to your male hormones, you'll be born looking like a female. Okay? But this is one of the more common ones in males, this 5-alpha reductase. You're born with a genetic defect in that gene. You can't make that enzyme right. You don't make the hydrotestosterone, and you're feminized, born looking like a female. Okay? Usually the testicles aren't descended, they're inside the pelvis, and the external genitalia looks very female. Okay?
So when you're born, if everything's looking pretty normal, how do they determine that things are not really right? Do they have to do testing? What would promote them to say, hey, something's not right?
Well, sometimes it's not even discovered until puberty.
Oh my goodness! Wow. Sometimes
it's not even discovered until puberty. But many times when they don't menstruate, you know, that sort of thing. And this is like 1 in every 2,000 births, you know. So it's common. I mean, it's not, you know, there's a good chance somebody on this campus has got an issue. But most of the time, it's more ambiguous. so that it's like, is that a female or is that a male? Is that clitoromegaly or is that micropenis? You know, and you can't tell because the urethra sometimes looks like a vagina and blah blah blah, and so, but it looks a little funny, okay, and so that sets off the genetic studies and the ultrasounds looking for ovaries and testicles and things like that. But usually it's best just to leave them alone, see how they grow up, see what sexual orientation they develop, and let them live their life the way they want to, instead of trying to do surgery and everything.
With the lack of the male hormones, will they also develop breasts as they enter that age? Or
No.
Okay.
No, they won't go through puberty.
Okay. So they just,
They'll
they're
look
completely
like
ambiguous through puberty.
Prepubescent male,
Okay.
you know.
Okay.
And so that's, you won't get, you know, growth of pubic hair and that kind of stuff. You won't grow muscles.
So completely ambiguous through puberty. Okay.
Yeah.
So if the testicles stay in the pelvis, do they become like, I don't know,
Very often.
can they become cancerous or?
Yeah, there's a higher incidence of testicular cancer with undescended testicles.
So can they still have kids?
So even what you would call, you know, a normal male can be born with undescended testicles, or just one sometimes. And they'll surgically take it out and put it in the scrotum because it won't be fertile because the body temperature is too high to let the testes work. Okay, that's why they're in the scrotum in the first place, because the temperature is lower. So we know a lot about these kind of defects. We know the chromosomes they're on. Like it says right here, that there's at least 50 mutations already known just for the 5-alpha reductase deficiency. Because these proteins are 800,000 amino acids, and all you have to do is hack it in one place, and you might get it not to work at all. Okay, the wrong amino acid here makes it you know, the protein curves in the wrong way, and it doesn't work. Okay, and so there's a lot of places you could hit it and have it not work, but the end product is all going to be the same. You've got a 5-alpha reductase protein that doesn't work, so they're all going to look like they have the same problem, but they may have different deficiencies, different. This is pretty cool. This is a species. Have you seen this? This is a species, the spotted hyena, where every one of the females has that aromatase deficiency. So they grow up masculinized. They're the most vicious ones in the pack. That's a female. Okay, that's clitoromegaly. The uterus empties through that. The urethra empties through that. They have to have sex through this. So you can imagine it's a little difficult process. Okay? But you can also imagine that once they get pregnant, they've got to deliver through that.
No wonder they're cranky.
10% of the first
No wonder they're so mean.
Yeah, I'd be mean too.
Oh, they run the males off. You know, and you have to wonder, evolutionary-wise, once this happened to one hyena, how did it get ingrained in the whole population? Because there had to be some sort of selection pressure to select for it. And the only thing I can think of is that the females were more aggressive, and they just kind of ran off the others, and they survived better. Okay, because that's the only thing I can think of. Because otherwise, a 10% mortality is nasty for a population. So, but anyway, this is a special purpose or an interesting sort of thing. I'm sorry?
Is the top one normal?
I think that's the female too. You just don't see the clitoris there, I guess. But they're bigger than the males. They're meaner than the males. They run the pack. Whereas the other types of hyenas, that's not the case. So it was a whole behavioral change once all these women got all that testosterone. Ambiguous genitalia, like I said, you can be born anything from looking like a regular male to regular female. You can have the clitoris can be enlarged, or the penis can get smaller. So the genitalia can look sort of like anything in between. And I'll show you some. These are 6 different male babies with a range of something called hypospadia. You'll notice that the opening of the urethra is not quite on the tip of the penis. So that's a very slight case of hypospadia. If it gets further on down, you can see now it's on the shaft. Now it's almost at the base. Okay, and now it almost looks like female genitalia. You know,
So this is a defect in that gene?
yeah.
Wow.
Yeah, this is feminization of a male. Okay, so so obviously, like if you saw that as a newborn baby, you wonder, is it a female, is it male? You know, you really can't tell. Here's some older ones, not babies. You really can't tell by looking whether it's male or female. Okay? So, but this is what we mean by ambiguous, right? And most of these people are just fine. You know, they have sexual relationships and all sorts of things. And so, you know, people can have fun in lots of ways. So, the big tragedy was in the 50s, they thought, oh no, we need to operate and we'll raise her as a female. And it turned out they made the wrong choice. And it was really nasty back then. Okay, so, well that was just my little reel on that. Now we'll get to that.
Next one's in, guys.
We're going to be covering, yeah.
So is that the same thing as a hermaphrodite?
No, true hermaphrodite has both male and female organs. And the only way you can really get that is to be a chimera, where most of the time it's non-identical twins, a male and female twin, where the zygote fuses and they have all the chromosomes of both sexes. So they'll develop ovaries
They
and
usually have a
testes.
twin?
Okay. What?
So they usually have a twin?
They are the twins.
They are the twins.
Oh.
That happens a fair amount.
The one that
There's-
is female.
There's cases, well, there was one just like a year ago where a woman had her kids taken away in California because they did a blood test, and the blood test, the DNA in the blood test said that she wasn't the mother. Okay? But then they did a swab of the cheek, and they got the other set of DNA, and it matched. So they knew she was a chimera. So that happens occasionally. It's one of those rare things that you'll hear about it. Okay, the first we're gonna cover most of chapter 3, or essentially all of chapter 3 for this test. So it's all about organelles and how they function. If you want to read, you're gonna have that week off to get through all that. But I'm going to try to keep it succinct and just do a few things with the slides. And obviously most of the test questions will be things I've talked about in the slides and things like that. I'm going to just try to hit the high points of the organelles and how they function and that sort of thing. The first part of that chapter talks a little bit about cell theory and how that evolved. They used to talk more about things like spontaneous generation and other types of topics like miasma theory of disease and all sorts of things. So maybe the new editions don't do that. But it's still on the syllabus for me to bring it up. So I'm just going to go through that pretty quickly and just kind of give you a feel for some of those old theories and ideas. And that may be all we'll cover today. I don't know how long it'll take here. So, but I told you I'd show you some electron micrographs of cells. This is a lot different than a light microscope. I mean, you can get thousands and thousands of magnification on these things. And I think everybody can kind of identify this organelle here. Probably nucleus, yeah, that is a nucleus. Little nucleolus. Sometimes you'll get a nucleolus that's on the cell membrane here, but very often the chromatin sometimes will bunch up and form little dark areas on the membrane as well. And these tend to be areas where there are pores, and so they're actively making RNA that's being shoved out the pore right there. And I'll show you some pictures of nuclear pores later on as we go on. But these are mitochondria. Here. They don't look like the sausages you're used to, but occasionally you'll see the crista crossing over and forming those membranes, and I'll show you why those membranes are necessary in the future. And so these might be just little vacuoles here or something. So a lot of ribosomes out here, and if you get much higher magnification, you start to see the microtubules and things like that. But this is a typical cell under electron micrograph. Okay. I just thought I'd talk a little bit about, the world has changed in a lot of ways in the last 150 years. Can you imagine a world where nobody washed their hands because they didn't know that there were germs? We're talking American Civil War time here, 150 years ago. People didn't even clean the saws when they were doing amputations of the soldiers. They just went from one to the next, and infections were just rampant. If you actually lived through the thing, it was almost a miracle. So that was a different world. And so just in the last 150 years, we've a explosion of knowledge on all this stuff. And this is one of the first guys that started tweaking the status quo, because the world believed in something called the miasma theory, that most disease was just caused by bad air. Okay? That's why you didn't want to live in the valleys in medieval Europe. You didn't want to, in Italy, I've done a lot of touring around Italy, and all the little villages are up on the tops of the hills and the cliffs and the mountains, and there was nothing down in the valley where you'd think it would be nice. But they didn't want to be near the streams and the swamps and things like that, because they thought the bad air was what got you. Because they had no idea that malaria was spread by mosquitoes. They thought it was the water that was doing it. So they drained all the swamps, and that made things better just because the mosquitoes were gone. Right? But you can see how that fostered their view of the world, and even even the word malaria means mal-aria, bad air. Okay, they thought it was just a disease of the atmosphere. But Pacini, of our famous Pacinian corpuscle, okay, back in the 1850s, he started getting suspicious, and he isolated the cholera bacteria. These were when the microscopes were just getting good enough to start to see these things, okay, because they had to be pretty good. You've done a lot of work now. You looked at Lake Elbert and everything. You didn't see any bacteria because they're too small to see at those powers. Okay? You saw the little tiny worms and things moving around, but you didn't see bacteria. They're much smaller. Okay? But he came up with the idea that cholera was caused by this bacillus, and then a few years later, Koch came up with his postulates and basically proved that it was, because he did some pretty wacky things with infecting people and this sort of thing. But the cholera epidemics were massive, and they still occur in the world. I mean, this is not ancient history. It's mostly, they had a big cholera epidemic in Pakistan after the earthquake. They had one in Haiti after the earthquake. And it usually has to do with unhygienic conditions and bad water. It's spread. Usually there might be carriers, people who aren't terribly sick, but they're spreading it in their stool. Okay, and if that gets in the drinking water or in the stream, everybody downstream is at risk. And you can affect tens of thousands of people all at once. And this thing will kill you within a day of dehydration. Okay, and I'll tell you why here in just a second. So we're working away from that miasma theory, finally getting into some kind of a scientific way of looking at disease. And I think this again is just kind of stating what I just said, so I'll skip it here for a second. Another problem was spontaneous generation. It still wasn't until Pasteur in the 1800s basically did his experiments that people really took note of the idea that there were microorganisms that caused problems. And if you could keep them out of wounds, and if you could keep them out of a meat broth or grape juice, it wouldn't ferment. Okay? And he proved that by saying it's not the air. What he did was he took a broth sort of thing in a glass and blew the glass out into this long, thin neck. So it was open to the air, but nothing could actually get in just by blowing in on the dust. Okay, and so as long as nothing got in, there was no rotting, there was no fermenting, there was no the broth wouldn't go bad, that sort of thing. So he proved it wasn't the air. He proved that there was something in the air, microorganisms, and that's where we got away from the idea of spontaneous generation as well, because they thought that the broth would just spontaneously generate these microorganisms and maggots and flies and all sorts of things, but they just came from the tissues itself. They just spontaneously generated from decaying matter. And like I say, 150 years ago, these thoughts were out there. This was medicine.
And crocodiles were formed from rotting logs.
Yeah. Which makes you wonder. Did anybody ever see a crocodile egg? So there were a lot of ways to disprove this, but people didn't think scientifically. They didn't think, what could I do to prove that or to show that it's true? And it has to be falsifiable. Remember, I have to be able to do a test that can either be, it either works or it doesn't. Okay, And there's a lot of ways you could do that, but people just didn't think that way. Okay, they were so ingrained in the old, wives' tale. So, Reddy was another guy who did something similar to Pasteur, and he did it with meat. Okay, as long as you didn't let the flies get to the meat, the maggots wouldn't be on the meat. Basically the same kind of thing. But he did that in the 1600s, but still the idea of spontaneous generation didn't take off because they didn't have journals back then. There weren't necessarily a lot of people reading scientific journals. And then here's Pasteur, just so we know what he looks like here. But he basically showed that fermentation doesn't happen by spontaneous generation. And this was the interesting thing with this guy Snow. There was a cholera outbreak in the middle of the 1800s in London, and again they thought it was the bad air down by the river, the Thames River. But he scientifically said, I don't believe this, and he calculated, or he noted all the individuals where they lived who got cholera, and it turned out that they were all around this one well. Okay, or it was a big part of the city, but the center of it was this well. Okay, so he was suspecting that was the problem. Something was in the water that was getting people. So he basically was the one who spelled the death throes of miasma theory when he was able to prove this was true. Okay, I'm just throwing this name out, Robert Hooke, the guy that I think is most famous for coming up with the word cell. If you don't know where that came from, think about the word jail cell. Okay, a cell was where the monks lived, in these little rooms in the monastery. It was called a cell. So when he was looking at cork and leaves and things like this, and so when he saw those little blocks like that, it reminded him of that, so he called each one a cell. That's where the word comes from.
Wow.
Okay, so that's back in the mid-1600s there. I like this guy, Leeuwenhoek. He was a real sort of, merchant class, but he got pretty rich from making microscopes, and he wouldn't show people how he did it, but he came up with this. These things were pretty simple because it was just a plate with a hole in it where light would go through, and you put a specimen over the hole, and then you had a lens, a tiny little lens that you looked at with the light source behind you like this, and he could magnify things though. He saw blood moving in the capillaries of animals, like in the wings of butterflies and things like this. He was the first guy ever to describe sperm with a waggly tail. And he basically was the one who put an end to the idea that sperm contained a tiny, tiny little baby in there. And then all you had to do was put that in the womb like planting a seed, and it would grow there. Okay, but he disproved that there weren't any little people in the sperm. Okay, so that was another good idea that they had come up with. He saw, like I said, he saw muscle fibers, and he saw lots of little animalcules, things that were growing in the streams and rivers around his house, kind of like what you saw. He saw all those little tiny filarial worms that you can't see with the naked eye, and he reported on these. He drew these pictures of these, and he was actually called to the Royal Academy in England because the bigwig scientists at the time didn't believe him. Okay, they were like, you got to show this to us before we're going to believe all this stuff you're telling us about this. And so he became famous for doing that. But he wouldn't show them how he made his little lenses. And he came up with a real interesting way of taking glass rods and melting them until they started to form a drip. Okay, and then as it forms a dropule, and then he cooled them, so he had this round drop of glass, and then he would polish those, and that's how he made his lenses. For some reason, that was the best way to do it at the time, and it worked. Okay, nobody else knew how to do that. But he also, you notice he found the sperm. I remember reading a couple of his papers because they're kind of interesting, just reading the old language, And he was talking about how he got his sample from the act of marital bliss, because he didn't want anybody thinking he was fooling around on his own. Koch's postulates. This is basically once germ theory got into the throes he came up with the idea that we need to have a way to prove that something is infectious, because not all diseases that come on are infectious. Some of them are just, say, late blooming genetic diseases, okay? Or there might be diseases of nutrition or something like that. So, but if it was an infectious disease, you had to be able to take a scab or a pustule or blood or something, some tissue from an infected organism, give it to another healthy organism, regenerate the disease, and then take that and show that it's the same stuff, okay? That was called Koch's postulates, okay? So he was sort of the first scientist of germ theory, right? So, and I'm not going to make any real fine detail out of these issues, but I'll list them here in the next slide, I think. Another thing, this is actually what's in your book, talking about cell theory, okay? All organisms are made up of one or more cells, okay? And again, let's get the idea. We all sort of accept it as true, right? But technically, cell theory is a theory, okay? I think, are we locked out?
Locked out?
So philosophically in science, we could prove that, well, no, it's not the cell that's the basis of life, and blah, blah, blah. You know, we all don't believe that's ever going to happen, but it's still a theory, okay? And a lot of other theories are so well-founded that we think they're never going to be overturned, like, you know, evolutionary theory and germ theory, atomic theory, all these things that have been proved so many times, but it's still a theory. And then cells are the fundamental functional and structural unit of life. In other words, if you have a cell, it can't do anything other than what its own proteins and genetic makeup allows it to do, okay? You can have multicellular organisms, you know. I can't do anything that my genome won't allow me to do. I can't make neurotoxin and spit it at you like a snake because my genes, I don't have the genes for that, because my cells don't have the genes for that. So everything I can do is based on, in some way, what each of my cells can do, okay? And then all cells come from pre-existing cells, and that's sort of understood with division and propagation. So that's just the very basics of cell theory, okay? And here's one that you might not hear about too often. It's called vitalism. This was a big issue for ages and ages, because everybody always knew that there was something special about life. You know, the stuff that you could pee out in urine, okay, boil it up, you could get these compounds and isolate them, and even the old alchemists knew how to purify these things and get crystals of urea, things like this. And these were always different than something you could find out in nature that wasn't alive. You know, you never found urea crystals in rocks, okay? It just didn't happen. So they thought there was something really special, an essence about life, and the molecules were totally limited to living things, okay? This was the first time this was disproved. Everybody knew what urea was. They really didn't know what the structure was, but they knew what it was. They knew it made crystals. They knew the melting point. And, you know, they knew a lot about it, so they could identify it. But it turns out this guy here, Wohler, made a mistake. He thought he was making, where is it here, ammonium cyanate, okay? Because he put the ammonium ion with the isocyanate anion here, and silver chloride is going to precipitate out. Remember, that was our test for chloride, our silver nitrate. Okay? And what's going to be left is the ammonium cyanate there. What happened, though, was he got a reaction between the ammonium attacking the carbon here, just like making an amide, and he got a double amide, and that's urea. Okay? This is the first time, by mistake, actually, that an organic molecule was made by inorganic synthesis methods. So it disproved the idea that you needed life to make these molecules. So he's considered the father of organic chemistry, much to your guys' chagrin, right?
Yeah.
But this was the first molecule, organic molecule ever made that disproved vitalism. Okay, so if you just have a one-sentence description of each of these things here, I'll give you some sort of true-false question or something that's easy, all right? Miasma theory is the theory that life generates from decaying matter. Okay? No, that's spontaneous generation. Okay? Something like that. Okay? So. We got a minute. Let me just. Okay, let's do it real quick. One second. Spontaneous generation. Life comes from decaying matter spontaneously. If that's not redundant. Okay. Circular reasoning. Miasma theory. Disease comes from bad air, basically. Cell theory. Cells are the basis of all function and structure of life. Germ theory. Microorganisms, whether they're bacteria, viruses, or other sorts of things, cause disease. Okay. It's not bad air. It's not curses. It's not witchcraft. Okay. It's not bad luck. Okay. Vitalism, that life has a special essence as opposed to inorganic compounds. Okay. So. Okay. And we got about 15 minutes just to sort of go through some of these membrane proteins here. Just to review, remember we have the cell. If we take a section of the cell wall, we've got that lipid bilayer. We've got our fatty tails in there, and our phosphate oxygen heads that want to be in the water. Okay. We've got integral and transmembrane proteins in there of all sorts of things. Okay. If we take a little section of that, you can see it's. Maybe we can blow it up a little bit more. We can see that it has those tails in there. And this is one thing I don't know if I've even mentioned yet. It's not just that any fatty tails can be in there. You will see a variety of things. But there has to, usually in our cells, we have a special arrangement where one fatty acid is all in the trans or saturated mode, and we have one that's in cis. Okay? The other, if it was a triglyceride, we'd have another one, right? Mm-hmm. But instead it takes that alcohol and the glycerin and attaches it to the phosphate, the phosphatidylcholine. Okay? This makes a really polar head that loves to be out here in the water. And now we have these two fatty tails. But this makes a big difference for our cells because remember, if we take animal fats and we put those fats together, they just lie like firewood, nice and straight, okay? They're going to solidify like butter, right? You don't want that to happen in your cell membranes. So all you do is you put a kink, you put a cis double bond in there, and now they can't solidify, okay? Now they won't wrap up together, and now you get a much more liquidy membrane, okay? So that would be a real problem if we didn't have that cis bond in there, okay? So that's the whole purpose of that. Okay, so here we go. Just another thing. The idea now, okay, this is, we talk a lot about it, but it actually has a name, okay? The idea that our cells are made of lipid bilayers with membrane proteins that can move around, just like you saw in that video, floating around in the surface, okay? Occasionally you'll get rafts that have a lot of cholesterol in it that makes them stiffer, okay? And apparently, just 6 months ago I was reading an article about how some of these membrane proteins won't work without cholesterol in the membrane, okay? Because they tried to make artificial membranes with these same compounds, put those proteins in there, they thought they were going to work fine. They wanted to test them and see what they were doing. They wouldn't work until they put cholesterol in the membrane. Okay? That kind of stiffens things up a little bit. But this model, this idea that this is how it all works, is called the fluid mosaic model. Okay? You could also call it the fluid mosaic theory. Okay, so what does all that have to do with me bringing up cholera in the first place? Well, in a way, it's because I want you to start thinking about disease as a dysfunction of cells, okay? And we're all, in our sort of civilian, naive way, you think of measles, well, that's a disease where you get red spots, okay? Well, now, if you're going to learn about medicine and disease and patients and treating these things, you got to start thinking about, well, what is causing those red spots? You know, what is going on at the cellular level that's causing all that stuff, you know? And so thinking that way, I think you'll find that makes this stuff a lot more relevant, okay? And thinking about how medicines work. How do antibiotics work? You know, they work in all kinds of ways. I mean, there are antibiotics that shut down ribosomes, antibiotics that shut down mitochondria, but they do it in a special way that they only get the bacterial ribosomes and the bacterial mitochondria, things like that, okay? Because obviously, if an antibiotic is going to shut you down, you don't want to take it, right? Those are called poisons, not antibiotics. And I'll show you a few of those as we go along. So it's just asking you to start thinking about in a biomolecular kind of way, and we'll talk about diseases. You know, I think you can understand by reading this why cholera is such a problem. Okay, these are cholera beds. These kind of things are flown in by the thousands when they have cholera outbreaks because they have so much diarrhea that you've just got a patient lying on this thing. The diarrhea runs down the pipe into the bucket. Right, there's a bucket. So it's just got a hole in the bed. Okay, and if you don't have IV fluids, you need to give them what's called the cholera rehydration formula. Okay, and since you know how to figure out molecular weights and molar solutions and things like this, when you go into an area like this, you'll be able to make the solutions for them. Okay, but they have recipes on how much glucose and how much salts and things like that you need to replace in these things, and you just got to get the patients to drink as much as they can because they are going to be losing like 6 liters a day in diarrhea, and that will kill you in 24 hours. Okay, so you have to do it quick. And they also give antibiotics now, but it's a nasty disease. Like I say, Haiti just had an outbreak after the earthquake, and it killed hundreds of people. And they also hung about a dozen women for witchcraft for starting it.
Oh, God.
Yeah, that was nice.
Wow.
Yeah. But think about what's happening here. You have a bacteria that has a toxin. Okay? It has. I can show you the picture of the toxin. I think. There you go. That's the structure, the molecular structure. These are all amino acids. It's all a protein. I don't even remember which part is which right now, but it has half of this molecule is an identification molecule, which basically fools the intestinal cells to think that it's food, basically. It attaches to the intestinal cells. They engulf it in vesicles, okay, bring it into the cell. The lysosomes want to digest it, and what they do, the lysosomes basically cut the bottom part off, which is the identification part, and this toxin is released into the cell. And what this toxin does is then float around the cell, and it grabs onto the chloride pumps, and it lets chloride just gush out into the intestinal fluid. And it's not going to go out by itself. It has to take a positive ion with it, right? Neutral has to be neutral. But so you're getting chloride and sodium gushing into your gut. That creates what? High concentration of salt. You've got a hypertonic fluid. That's going to suck all the water out of your system into your intestines and right out your rear end. Okay? So that's what causes the diarrhea problem. It's because the cholera toxin is attacking the chloride pumps in your intestines. Okay? And until that's washed out, until that's gone, you're going to have to rehydrate these people. But that's what it's saying right here. Osmosis is the problem because of the chloride. Okay, so. Oh, I can ramble on. I'm going to let you go 5 minutes early. So we got plenty of time to cover things.